All right. Well, thank you all for joining the GA4GH Phenopackets AMA. Ask Me Anything. I'm going to hand it over to Mallory Freeberg to introduce the session, and we'll spend a little bit of time giving a very brief intro, and then mostly want the session to be all about questions and answers. So thank you all so much for joining. Mallory?
Thank you, Benjamin. Hi, everyone. It's my pleasure as co-lead for the GA4GH Implementation Forum to welcome you all to this is the second Ask Me Anything event for GA4GH standards. If you missed our first event last month, it was on Beacon, and all of the recording and materials and slides from that event are available on the GA4GH website. But today we are crashing the Phenopackets meeting and hosting an Ask Me Anything all about Phenopackets. So let me open my slides. So for those who aren't familiar with what an Ask Me Anything or AMA is, basically this is a more informal meeting. So we're going to be very light on the presentation, very heavy on the people asking questions. So it's anyone to ask any questions about implementing Phenopackets, maybe share your experiences. And thankfully, we have a lot of Phenopackets experts on the call today. We'll probably end up both asking people to raise their Zoom hand, and you can verbally ask a question, and we'll have some sort of discussion based on those questions. And anyone's actually welcome to answer. It's not just us asking one or two people. If a question's asked and you have some experience or some feedback, please feel free to raise your hand and answer the question. Also, we probably will get a lot of questions in the chat as well. So that's what happened with the last AMA. So if you have a question, you'd rather type it, feel free to use the Zoom chat. And then if you want to answer that, also feel free to use the chat, and we'll capture all of the verbal and the written questions and make those available as well after the call in sort of an FAQ document on the GA4GH website. So we'll try to get to as many questions as possible. So if you're raising your hand and waiting a while, apologies, please be patient. We'll try to get to your questions. And we also will follow up on that Q&A or the FAQ document afterwards as well. So people will continue to answer after the call ends. Oops. So before we get started, I wanted to ask everyone on the call, I won't call on you, so this is a safe raise your hand, but if you have ever tried to implement or have implemented Phenopackets or have been involved in the development, just ask that you raise your Zoom hand, or you can also raise your physical hand. Just want to get a sense of who on the call are experts today. Oh, excellent. Some physical hands and some Zoom hands. That's great. So as I mentioned, anyone is welcome to answer any of the questions, or if you have input or feedback on the questions asked, please feel free to raise your hand and answer. So we'll go back to, whoops, 1 second. Ah. 1 second. Go back to my slides, because what we're going to do next is have a very brief, although I said this was presentation light, we're going to have a very brief 5-minute update on Phenopackets. So I'm assuming everyone here sort of knows about Phenopackets or have worked with them, but we want to sort of hear from the developers, just sort of a quick update on some progress. So I will pass off to Jules, and Jules, I am happy to advance the slides whenever you need me to. Just give a shout.
Okay, hi, thanks. Yeah, so we're not got a lot to say because we're trying to keep this stable standard rather than a fast-moving, changing standard, because that just breaks everything. So I thought we'd just sort of keep an update on just so everybody knows where to find all the information, and then just a very quick overview, just so that in case everyone's kind of forgotten what it all looks like. So hopefully everybody's seen all this before. The schema itself is now a version 2 of the schema, which was a bit of an upheaval, but depending on which parts you used, it wasn't so bad. So we're just going to work from sort of top to bottom, and really the idea of this is for tying together genomic and phenomic information. So that's the kind of the key part you need to remember about this, and it's also individual level data in a sort of a single file-based format. So that file-based format is the Phenopacket, and that's the individual level data that you can see there in black. So moving up again, we've got a family and a cohort. So a cohort is just a collection of Phenopackets based on whatever you fancy grouping your individuals together with. It may be a particular disease or sort of certain characteristics, or really anything you want. Then the family itself is really meant to be a family of genetically related individuals. So this is why they have a pedigree, and then links to various bits of information you need to do some sort of cohort and family-based filtering for genomics with that. If we're moving down, we got the Phenopacket itself has information about diseases, about that have been annotated to that person, interpretations for the genomics of, so this is how, if you're going in for, say, genetic testing, the interpretation may be, well, we actually found this particular variant, which is implicated in this particular disease in this gene, and that's sort of how that all gets folded together. Biosamples are there more for kind of cancer style things where you might take biopsies and then measurements on the biosample, and then we've got the phenotypic feature, which we've got in light blue, and that's kind of the really sort of one of the key parts of the Phenopacket to actually describe the phenotypic features of a patient or of a biosample. And then lastly, we've kind of got things like medical actions where you can actually also put in there what's kind of treatments that may have been given to the patient. So if you move on one more, conscious we've not got a lot of time here, so I'm just going to just go through, rattle through some links of where you can find things. So if you don't know about the actual formal specification of the Phenopacket, so that you can find that on the Read the Docs site. So that's on phenopacketschema.readthedocs.io. So start there if you don't know anything, or if you do know things, to keep going back there, because that's where it all sort of really is all well defined. The reference implementation is actually defining the schema is written in protobuf, and that's in the Phenopackets repository, Phenopacket schema repository, sorry, of the Phenopackets organization on GitHub. So the benefit here is that we can then generate language bindings for pretty much any language that you might be interested in using, but straight out of the schema itself. The slight downside of protobuf is that although it's very nice to actually define a schema so you can get syntactically correct stuff, information, you may not have semantically correct information. So this is where you need the validation that goes on here. So we have a JSON schema included as well. So I know there's a lot of people have requested a JSON schema of the Phenopacket schema because they don't like protobuf because it doesn't quite gel with them. So we're using JSON schema to do the validation, and that you can find in the Phenopacket tools. That's a Java library, which is wrapped in a CLI itself as well. So you can use that in pipelines if you need to do some validation on large amounts of Phenopackets. Again, this is very much a kind of rare disease focused thing. So it uses the HPO as one of the ontologies for defining the phenotypic features. So we go next slide, please. So this is really Peter Robinson's work. So I think he's on the call at the moment. I don't know, Peter, if you want to talk about this, or I'll just say what I can, say what I know about these things.
Hi, everybody. Yeah. So yeah, we wanted to create a resource that would enable other software packages to show the full value of Phenopackets. And so, before we just had whatever 5 or 10 examples, but here we curated by hand over about 2.5 years, a total of now 8,000 cases with over 500 different diseases, and these are derived from publications, so it's fine to use them in whatever analysis. And we've now used this for I think 4 different analysis projects that we could show, and they're freely available.
Thanks, Peter. And that's really it. I just want to sort of say that's kind of the state of where we are at the moment. And really, it's time for people to ask some questions.
Excellent, and great job keeping to time. I didn't have to budge you at all to keep to time. I realize it's a very, very short time, but we do want to have a majority of the time for questions. So that is this time. As I mentioned, we'll start with some verbal questions. So as David is demonstrating, please raise your Zoom hand. But if you would rather ask your question, or before you forget it, put it in the chat, and we'll also be monitoring the chat. Beatrice will be helping me, and we'll be picking some questions in the chat as well. And then if you have any response, no matter if you're an expert or if you're new, your input is valuable. So please do raise your hand to answer. And we will kick us off. David, I see you got your hand in first, so feel free to unmute yourself and ask your question.
So do we want more like a, I guess, specific questions about Phenopacket first, or like if I have questions about the vision, the future, and those things, can we start with that, or
it's first come, first serve. So whatever question you would like to start with, feel free.
All right, sounds good. Thank you. So I work in a project that has invested a lot in Phenopacket. Okay, like we have a platform that ingests data as Phenopacket documents, that organizes data like following more or less closely the Phenopacket model, that exports clinical data in Phenopacket model and those things. And so it has enabled us to identify like, well, first of all, that like it's a great fit, right, to organize. It's a great format as a data exchange format for clinical data, information about biological samples. And then we're starting to see links with other GA4GH groups and studies, like in experiment metadata. We see that it is a logical project progression of I have a participant, a biosample. There are experiments run on those biosamples. So like, so all of that to say that like in the past some years where we've been using Phenopacket, we've identified also like a couple of gaps that we've raised here and there. You know, like maybe I can get out of the dust this old Easy Retro board that was for the Ascona meeting in Connect last year, where like we listed a bunch of things about Phenopacket, like a specific question. We feel this thing is missing, and we never really had a chance to discuss these and try to address how, like, basically we could maybe do a next step for Phenopacket, maybe a 2.1 or something like that. So like I'd like to know what is the vision for the next year or 2 years for Phenopacket. I understand there's a meeting once in 2 months that got brought back just recently. I think we met only once up to now, but like I want to be sure that there's a real desire to bring things beyond and maybe improve on places where some gaps have been identified.
Great question, David. And Moni, I feel like you want to respond.
Yes, I would like to take an opportunity. Sorry for being late. I'm Monica Munoz Torres, and I have the privilege of co-leading the work stream on clinical and phenotypic data capture with Benjamin, and have been working very closely with Peter and Jules over these years. I think that the most important thing to think about, David, is what do people need, right? Making sure that anything that happens with our very stable, broadly distributed standard is an advancement and an evolution based on what the community is identifying as the next step for what the plasticity that Phenopackets offers, what the utility that Phenopackets offers. We can sit down to try to spec, you know, every single corner of the standard until the cows come home, because this is what we think would be best to represent a few things. But without a driving case and without the users asking for those improvements, then I think that we would be, you know, we would doing ourselves a disservice. The work has taken us to this place, and I'm not saying that it's perfect. We all recognize that there is room for improvement always. I'm just thinking that we really should base this on the foundation that the community approaches us with a need and for improved and enhanced, more robust representation of phenotypic data or the clinical data. And then when that happens, we'll start working on what that improvement could look like at 2.1, at 3.0, but it needs to continue be as it was from the beginning, community driven.
Thank you very much, Mondi, for the answer. And I think maybe like a way to get these kinds of conversations started. And as I was saying, and I know there's a once in 2 months meeting that's just getting restarted. Like instead of making these like just like presentation centric, like I have a project using Phenopacket, there could be a corner that's like really like, let's get into the details. We feel that the connection to this thing is missing. Maybe there's this category of field that's not there. It's a place, because you know, like if we throw that on the G4GH Slack channel, if we put it, things get lost because like there's little messages everywhere.
Yeah,
You bet.
David,
You bet.
Totally agree. I think this is brilliant to have feedback. And Moni, you're right. We want to keep things so that it is, you know, really applicable and really, really driven by people's use rather than just like, well, we could add it, so we did. Because, you know, you never know what might end up in the end. It's probably something you didn't quite want, or didn't fit someone else's use case. So having an actual use case is incredibly important, and we have to have that. Isn't in terms of where you end up putting these things, I agree that having just presentation things is not the ideal sort of thing. This is definitely a good forum to start asking this sort of stuff. But the most concrete thing you can do is to actually go to the GitHub website, go to the GitHub repository, the Phenopacket schema repository, open an issue with precisely what your problem is, and then hopefully even something to sort of like an idea about, and this is how we solved it, or how we would like to solve it, or how it would be solved. And then, you know, other people can actually see this. We can then discuss these things at one of the meetings, and I think that would probably be the most expedient way of getting things to where people want it. So yeah, we have to be driven by users and also solid use cases, and then actually, you know, spec this thing out and come to some sort of agreement on it. So that was one of the links I gave you. I'll put it in the chat in case anybody doesn't know where to find it, but that will be my answer. David, does that help?
I
Yes, it
pasted
does.
it there.
Sure.
Oh,
Great.
cool. Thanks, Manny.
Yeah,
Thank-
it helps a lot. Thank you for the answer.
Cool.
So
Thank you, Jules and Moni. Peter, do you want to respond to this question as well?
yeah, I mean, I completely agree with what Moni and Jules were saying. I would like to say, so we have identified two things that we think definitely want to be added. For instance, in the phenotypic feature, there is an age of onset and an age of resolution, but there is not an age at, we observed it now. And so for things that are repeated, like seizure, it's hard to use. That would be a non-breaking change, so you can just add that to the model without sort of breaking everything that came before. Another thing is that there's no way to relate a measurement with a phenotypic feature, and that's sort of underspecified. And so those are the two things that have occurred to me over the past, since the standard was published, that are just wrong or not, or just need fixing. We have heard that we have gotten issues. People have told us things. I would like to suggest, right? I'm not sure if that was. I think that's been planned anyway, to have some workshops where basically people say, oh, kind of like Phenopackets, but it would be more useful for me if it also had this. And you know, so I think what GA4GH needs to decide is, does this want to go in Phenopackets, or is it a separate standard? So for instance, things like environmental exposure are super interesting. They're also very difficult to measure, and maybe that it's not, I'm not taking any opinion, but maybe that's not ready for going into Phenopackets. There are also things about, you know, a population background, or, you know, race, ethnicity, that are super controversial, and where our initial thought was, well, it's just not ready yet. But we'd like to, you know, discuss things like that, where there are probably many different opinions, and if there's a clear consensus that we should do something or we shouldn't do something, that could inform version 2 and version 2.1 or 3. And so I'd like to propose maybe that we set up some meetings where people actually come with their examples, and we really get into the weeds. And you know, we can start that off with issues on the GitHub tracker, but I think we really need to have some one-on-one Zoom time to get into the weeds.
Thanks, Peter.
That's anything stupid, Jules or Moni? Not too bad. All right.
I'll just make two quick points on that. I think that all sounds great, and perhaps a G4GH Connect session could be another venue to do some hands-on use case exploration. We have the upcoming one in Uppsala in October. But also with our new GIF co-lead starting, we'll have a bit more capacity in GIF to enable potentially some more sort of hackathon in the weeds type events. That's something that we're very interested in, exploring what could be helpful and useful. So if it's helpful to reach out to GIF in regards to doing something more hands-on that's not a part of Connect, for example, I'm very happy to chat about that in the future.
And Anne just shared a really good comment on the chat, so I want to make sure that it gets picked up as well. And we can welcome you to join us on the next Phenopackets call as well, so that we can capture those. It doesn't have to be a meeting. We can start with a Slack conversation or an email, but let's make sure that we capture that on the next Phenopackets.
So yeah, I'm actually working with the researchers on the exact plan that's funded by the Office of Women's Research, the Office of Autoimmune Disease Research. And Peter, this is with, it's so lovely to see you, by the way. This is with Richard Schurman, who is the scientific director at the National Library of Medicine. We work with Chris Mungle for ontologies. And we're doing a lot. LifeBit is spearheading a federated data analysis, and we're working with ArcGIS for environmental exposure, capturing also the social determinants of health detail and layering it in a GIS layer. But because it's patient data, data doesn't leave each of the different nodes, because you can't, to a certain level, get to that level of detail. You can only aggregate up for sharing that. So I'd be curious what your thoughts are, or at that meeting, where would Phenopackets play a role? And I think it's a huge opportunity for us at the Office of Autoimmune Disease Research and the National Library of Medicine to do something that could demonstrate. But yeah, so those are just random thoughts.
Thank you. And I think it's critical to share a lot, in alignment with this, as we just shared on the chat. We just started human exposome standards study group within GA4GH that launched within the ClinPheno workstream. So let's make sure we involve you and your colleagues, Anne, and everyone who's interested.
Yeah, so that would be Vicky Shanamagan, who's the inaugurating director for the Office of Autoimmune Disease Research, which in the US was congressionally mandated. And it's really freaking amazing. We've survived all the heck that has gone on so far. So anyway, we are still standing here despite everything.
We wish you much strength during this time, of course. Yeah, I think there's a kickoff meeting for the Exposome Study Group June 6th, if I remember that correctly. So yeah, that would be a great way to align as well. Okay. Any other comments on this sort of question around the vision for Phenopackets and ways to bring in use cases? Otherwise, we can move on to another question. Go ahead, Michael.
Just from my point of view, just want to reiterate this. Some things are not properly defined and could be better defined, as Peter said, probably by some general resolution in GA4GH and the race, population background, and so on. I just wanted to write it, but you're faster. We just have the same discussions at Beacon, where we know that the fields we are using, they are orthogonal, and can be differently interpreted. We found in our databases that we use a compositional model, for instance, for population background, where we do estimations based on models and reflect them, and so on. So there are several things, but for lots of population and for lots of epidemiology things, especially from the US, you need kind of the race label and so on. So that we should have a general agreed upon way to use the right vocabulary and the right data structures, and then refer to some explanation why it is done that way. And this should be like a general kind of GA4GH thing to resolve.
From what I remember, and Orion, I think, will be able to give more information on this background. Was the pedigree group was looking into the race, ethnicity sort of issues and what was around that and how to represent these things. And we had a presentation by I can't remember what the lady's name was. But the upshot was that there were so many different ways of slicing and dicing this information, and also completely inadequate ways of actually capturing a lot of the information as well. That it just wasn't really ready for any kind of standardized way of actually recording it. So if for example, if you look at, say, the British sort of ethnicity is completely different from the way that the American system records their ethnicity, just in a kind of what's the words, either in like healthcare systems or in sort of like government systems. They're just completely separate things. The way they record them is different. The actual values are different. We can't cope with every single different country's means of doing this. So we have things like Nomad, which on the other hand, is going sort of into more kind of trying to look at distinct populations, which we could start trying to use those codes as well, because I think they're a lot better. But in terms of there is at least something there where people can agree on. But that doesn't capture everything that everybody wants. So this is why I think we just basically kind of raised our hands and went, I don't know, this, like, whatever we do, we're going to make a mess. So I'd rather not have the
exactly. Same on our side. We don't want to define the standards, how things are reported, but somehow there's a need for asking for how does this correlate, so we want this label and so on, even if we know that the label and so on. So we need some general guideline for the technical representation and for the yes, we mean this when we say this, and then wrap it in some code, right? But that should be a group. Should it be?
I feel like that could be a whole group on its own.
Yeah, is it clean pheno, or is it something? Probably something even more, because we have the ethics and so on it. So something to bring up at GA4GH. Good thing for Uppsala maybe. So, but I think we should move on. We don't want to get caught on this here right now.
No.
Well, the discussion is very exciting. But yes, let's see if anyone else has another question. And while people are thinking about their questions, I asked a question. I'm curious, people who are on the call who maybe are starting their implementation journey or are currently implementing Phenopackets or thinking about how to, basically, what is your use case? Why did you choose to implement Phenopackets? What problem is it solving? I'm curious, like, what people are actively working on now. Oh, I see some chat answers. Anyone who has answered in chat want to unmute themselves and verbalize their answer. Orion or Victor?
I don't think there's anything else. I was just following up with what Jules was saying, but from the pedigree working group. So the resources there were Ancestro and some of the work going on in the ClinGen Ancestry and Diversity Working Group. But
yeah, I'm sorry, Mallory. I was concentrated writing my message. What was the question?
Oh, it's just about what are your use cases that you're currently implementing Phenopackets for? What challenges are you solving with it? But I think you would answer. Go ahead.
Yeah, so we already have implemented, and so yeah, about last year, we have a Django service that we use with a Postgres database to ingest and store Phenopacket data. And so yeah, around last year, we migrated from Phenopacket v1 to Phenopacket v2. But yeah, so we're as we're gathering new projects that are using our services, we have new use cases, namely one for agricultural and environmental data, where some samples or biosamples are sampled from, let's say, a river or a farm somewhere, and the geolocation of the biosample is of interest for the study in question, and would like to capture that information. So yeah, so far what we've done is to extend our biosample schema, our extension of the schema in our implementation to store that data. But yeah, I'll be happy to open an issue in the repo and to see if other people have similar use cases and want to use that. We also have another one, which is so the variant interpretations. Yeah, I don't remember the full hierarchy, but yeah, we'd also like to have to express variants of interest in situations where we don't necessarily have a disease attached to it. But at the moment, you kind of need to have a disease to include a variant expression or a variant interpretation. So that's kind of forcing us to create a kind of mock empty diseases sometimes, and it becomes a bit difficult to interpret the data after that. So those are two use cases we have, and yeah, we'll transpose to well-written GitHub issues with our use cases. But yeah, if anyone has similar interests, we'll be happy to chat with you and yeah, see if. Yeah, we use GeoJSON as well. Yeah, I saw that in the comment.
Nice, great. Thank you, Victor, for sharing your use case.
Sorry, please put those on the in the GitHub issues as two different things. I think there may already be one GeoJSON or some geolocation information already. So if it's in there, then can you just sort of continue that one? Then we can keep track.
Okay, perfect. Yeah, I'll bump it up. Sounds good.
We need a little like put it as a GitHub issue button.
Yeah.
Great. So I'll go through. I see some hands raised now. So Kais, do you want to unmute yourself and either share your use case or ask a question?
Yes. Hi, everyone. So do you hear me?
Yes.
Okay. So currently we are working in the frame of a project called AfriGenD, or African Genomics Data Hub. And within this project, we are interested to genotype phenotype correlation. And currently we are investigating Phenopackets to see if you can use it for this kind of sub-project, so to correlate genotype and phenotype information, and what kind of input we can use with Phenopacket to have this kind of correlation. So maybe we need to know more about it and to hear from you if we can use it for this purpose or yeah.
Great. And what was the name of the project you said again? I missed it.
AfriGenD, which is African Genomic Data Hub.
Okay.
Thank you.
Perfect. Thank you, Kais. Dave, you're next with your hand up. Go ahead and unmute yourself.
Sure. Hi. I'm Dave Gordon. I'm at Nationwide Children's Hospital. Our use case is in the clinical space. So we've been looking at ways to kind of consolidate both manually curated phenotypes through our genetic counselors, as well as looking at clinical notes in Epic, pulling out phenotypes from there, and developing a system to kind of maintain and corroborate those together along with genomic data. We're looking at capturing evidence along with the phenotype terms. The project we're working on is called Phoebe. It's a network graph representation of that evidence data along with the phenotypes. And we're looking at using Phenopackets for interaction between that and other tools, as well as being able to export, say, cohorts that we find within our clinical data and research data. That's our plan, to use it for that interaction.
Great. So the export with cohorts, are you like sort of wrapping all of the Phenopackets into like a larger structure, or just representing them per individual and then calling that the cohort?
That's a great question. I think it may depend a little bit on the use case there. Internally, we've got our own representation of it because we're capturing some ancillary data that I don't think is in the schema for Phenopackets, nor does maybe it belong there. I think it depends is the answer. whether it's truly like the cohort in the Phenopackets schema, or whether it's more like individual Phenopackets per patient. I think it'll depend on the use case there.
Yeah, it just reminded me, I think there's an active study group in G4GH about federated cohort building, and that just sort of made me think about, I think that is focused on like bringing in information from other data sets into like a virtual cohort, but this kind of reminded me of that work.
Oh, that's great to know. Yeah, that we'd be interested in that sort of thing, I'm sure.
Yeah, there must be information on the G4GH website. I don't know off the top of my head, but I think they're still active. Great. We'll get to Orion, and then we'll get to some of the stuff in the chat. So Orion, go ahead.
Thanks. Sure. I was just going to share that we have implemented Phenopacket early on within the Phenotyps tool, which is a software platform for managing structured phenotype and family history data, usually in a clinical genetics workflow, but occasionally for research projects as well. Found it very helpful as an additional export format, in particular for both patient records, with the structured phenotype and genetic data, and for exporting families. So all of the individuals with the structured phenotype, genetic, diagnostic information for them with the family structure. So that ends up being used at a lot of our customer sites for moving data from phenotypes to downstream analysis systems, usually the ones that are doing the variant prioritization systems like Imagine, or other tools, just as a very convenient standard way for that data to move downstream. So similar to what I think Dave was mentioning at Nationwide. So.
Great, thank you for sharing.
Yeah, thanks.
I see Nicholas Owen, you put in a use case in the chat. I don't know if you wanted to unmute yourself and describe it verbally. If not, I'm also just happy to read it out loud. It sounds like you're exploring Phenopackets and other GA4GH standards for sharing human disease information across research silos, yeah, without sharing the underlying personal identifiable information. That's great. Did you want to add anything to that? I also realize not everyone is able to unmute themselves all the time. Okay, great. Anyone else have any use cases they want to share or projects that they're working on? I mean, you have the Phenopackets experts on the call, so now's a great time to talk about what you're working on and maybe get some input from people. Or if anyone who's a Phenopacket expert wants to comment on any of the use cases they've heard, things that they think could be next step to start exploring for support. Happy to hear any input on that as well. Go ahead, David.
Oh, okay. While maybe some people are brainstorming on your last question, I was wondering, because I know GA4GH does a landscape within driver projects periodically and those things, and I was curious right now whether if GA4GH is aware of how many driver projects actually really use Phenopacket. You know, not just saying yes, we're thinking about using it, but actually doing something with Phenopacket, making their data available as Phenopacket somewhere, or those things. We'd love to find other resources online that have their Phenopackets ready or available in a way that could be shared, or those kind of things.
Yes, I was going to call on you, Angela, if you wanted to take this one.
Yeah, just really briefly, I do have that information from the driver projects who responded to the most recent survey, and I will pull that in the background and share that with you. But to your point, it's relative. Like, you know, the question is, have you or are you planning on implementing? And the answer is yes or no. So we don't have the deeper details on sort of what that really means in practice, but you will know, you'll have a starting point at least.
And also to add, I think this is related to some discussions going on at GA4GH about like registries and sort of having a centralized place that we can link to where actual implementations, reference implementations, example implementations exist. I think the struggle right now is basically how we scale that and make it sustainable, because sort of asking people once a year to update is fine, but it doesn't really get you like the latest and greatest thing. So this like registry idea I think is great. I think we're just working through who does it, how is it done, how is it maintained, but that could be, I think the idea is that gets rolled out for all of GA4GH, and you can start with the very technical standards, which may be easier to link to implementations, but Phenopackets, you know, could be a prime user of that sort of service once we have it. Benjamin, I think you had your hand up next.
For folks that have raised use cases already, or others, I'm curious how people are generating Phenopackets, right? Are they being automatically generated through some scripts from an EMR? Are they being converted from another format like FHIR? And is there desire for that?
Good question. Anyone want to raise their hand and give an answer? I don't know, Peter, did you have your hand raised for something else?
Oh, sorry. Related answer, I guess. I mean, I think the value of Phenopackets will come once a lot of software has been written. And I remember doing an exome before VCF and kind of having to do everything by hand. It took us about 2.5 months to do one exome. I think we're still at that stage with phenotype data largely. And so I think for genomics research, what we know about right now, a bunch of groups have adopted Phenopackets as an input format for diagnostic software. So we have for Exomizer, for instance, we know of a bunch of software for converting from FHIR or OMOP to Phenopackets and back. We've been developing software that essentially starts off with a collection of Phenopackets, does genotype-phenotype correlation analysis, clustering, and a few other projects like that. And I think as that starts to come out, then people will be motivated to use a format like this, because then they can use all of this software. And so my request is, if GA4GH leadership knows of people doing this, let's get together and let's say, what are your goals? And can we maybe collaborate on some of the software? Essentially a lot of the downstream analysis is going to be whatever, a t-test or a chi-square test. But if you're using ontologies, it's not quite that simple, because you have to take into account the inheritance on the hierarchy. You have to take into account missing data and things like that. And so there's a lot of potential to really automate this kind of analysis and make it really easy to do a lot of very sophisticated things, but we need to have software to do this. And so it would be great if people are working on this, that we get together and don't reinvent the wheel more than necessary.
Yeah, this reminds me, or sounds to me like a potential for something we call a GIF project, a GA4GH Implementation Forum project, which there's information about what that is on the website, but essentially it's people working towards the common goal, and they sort of have the support and they're doing the work already, but then they kind of get together under this umbrella project and do it together and have some outcomes. So that sounds like potentially an idea for a GIF project, which we don't have a call open right now for those, but likely later this year we will have another open call. Yeah. Victor, I saw you had your hand up, if you still want to say something.
Oh yeah, it was just to answer Benjamin's request for examples of where we get the Phenopacket. So in our case, in the Bento platform, let's say we're supporting different projects, and they store their data in different formats. So sometimes it's going to be a database that we have to connect to through a VPN and run a script to convert the tables to Phenopacket schema and ingest that in our services. Other times it's going to be CSV files, or we have tons of input to output. So over the year, it's been Julian, one of my colleagues, that we've been making ad hoc scripts to do that process of taking a source format and mapping it to something else. We're currently, we started a ETL application for us. So we'll probably try and make it as agnostic as possible, for hopefully making it useful for others, not just our use case, but that not in the short term, that's for sure, maybe in the longer term. But yeah, we're actively looking at ways to better organize how we convert from various data formats to Phenopackets in a single service that doesn't force us to scatter around scripts. And yeah, so something we're working on for one of our projects. But yeah, I would love to know how others have implemented conversions from disparate schema formats to Phenopackets. Would love to hear how others have implemented that. So yeah.
Yeah, thank you, Victor. Yeah, I see there's a couple of chat comments related to converting. So Joshua, it sounds like you're looking to see if anyone has use cases for converting to another format like TopMed to Phenopackets. Is this something that you're working on?
Yes, I don't know if my connection is stable and you can hear me okay.
We
But
can hear you.
I'm in a remote traveling. But yeah, we're converting from TopMed to Phenopackets, and we're building the tool to hopefully be able to convert from other data formats as well too. And we're getting ready to look for some groups that could beta test what we're building.
Great. Hopefully some people on the call who will reach out to you. Thanks for sharing your email. They can connect with you. Let's see. Anne, I also see you have a comment. Did you want to unmute yourself and verbalize it?
Sure. I just heard Peter say that he's converted it from OMOP to Phenopackets, and we have everything in OMOP. That's the and so if there are some things that I could beg, borrow, and steal, we would wrap it into a Nextflow workflow. I think we work with single cell data, so we're working. It's a cell knowledge base that the National Library of Medicine is focused on, using experimental data to connect to ontologies and to derive novel insights in terms of connecting diseases to drugs in unknown, previously not connected, but it is a demonstration pilot to do federated data analysis. It's the first one within the walls of the NIH, and shooey, lots of IT departments that we've talked to. I think it's 4 that we've had to go through. And as it is, it's a lockdown pilot, so not the full federated, but next year we'll go beyond the walls of NIH. Fingers crossed, but very interested in knowing what Peter has.
That's exciting. Beyond the walls of NIH. Michael, you have your hand raised. Go ahead.
I just wanted to make the point that if one does look for conversion from some data format towards Phenopackets, then it's kind of, you're in the same area like when you're doing conversions for Beacon. So also Jordi's group at CRG, they have done a lot with, they have done OMOP mappings. So basically put the Beacon and then a layer in between on OMOP stores to beaconize them. And that would be the same. And then wrapping, I mean, Phenopackets and Beacon, this is just mostly, don't get into the details, but it's mostly a little bit of rewrapping and remapping of some of the parameters. And then there may be some which don't exist in the one or the other side and so on. And this is actually something where it would be good to have an active alignment process ongoing. We had this before, just before the Beacon v2 verification process. We did a lot of, let's just take the stuff from Phenopackets, not completely. So if you do the one, then the other, and everything is covered in the GA4GH environment, and you're helping driving this. So I think this is just a good synergy.
Is there a place or a meeting or a line of communication where that kind of work is happening, or is it kind of done now that the V2 is out? It's kind of okay. Because it sounds like there's a lot of interest in this idea of converting and mapping, and lots of people having different efforts, and it'd be nice to sort of maybe have a single place where that's being discussed. Or Benjamin, maybe you have ideas for that?
Yeah, I'd say the ClinPheno meeting would be the right place to discuss that. And then if we need to spin off a specific work stream for a particular effort, we can do that. But right now we're trying to figure out how to prioritize and whether there's appetite, for example, for a FIDO Phenopackets, or to take the existing work or an OMOP to Phenopackets and either publish it as an implementation guide, or potentially even as an API or a Python library that could be published.
I gotta say, there was part of the Biohackathon 2020, I think it was. I created a Phenopacket, OMOP to Phenopacket exporter, so you can link it to an OMOP database, put in a patient ID, and then it'll pull all the data out and put a Phenopacket from that. So I'll put the link, I'll relink it in case anyone didn't see it in the chat. If anyone has any more to add, it's pretty basic because it's just something we did in a week between a few of us. So it's not like it's all singing or dancing, but it's sort of got all the basics there. But if anyone would like to take that and then make it, more useful or productionize it or something, then I think that would be hopefully a good place to start on. At least we can, as a community effort, if people can put some pull requests in there or something, then that would be ideal. I think half the issue with OMOP is going to be the mapping of the ontology terms from that into HPO or whatever. But I mean, that said, that you can have a Phenopacket without using HPO. That's also totally legal.
Thank you, Jules. Anne, go ahead. Oh, you need to unmute yourself.
Sorry, you'd think after all this time we would all know, huh? But I was just thinking, so for this exposome project, we have everything in OMOP, mapping it to Phenopacket, and then what I need to know is that, what am I going to be able to gain? Remembering that I have single cell data. If I have it to Phenopacket, I have to try to motivate where I would go with this in terms of, we are connecting to some GWAS data, so we do get to the variant level. So and we're doing something called topic modeling to try to discover new endotypes. So I don't know, if anybody has ideas there, but I don't know what ClinPheno is, and I'm just curious where the Office of Autoimmune Disease could jump in, and this is a federated data analysis project as well. Is there a meeting in a year's time? Sometimes this is more fun to do face to face if it's possible. We're headed to a hackathon in June with Kati Berner. I don't know if you guys know her. She's part of HubMap, and she's got something called functional tissue units, which are anatomically correct. And we are going to connect it to our cellular information, what Richard Schurman has. He's behind NS4, necessary and sufficient markers for cells, so at the cell level. And again, it's a knowledge graph is what we're ending up with. So just great to convert, great to connect, even great to connect to Beacon, but you have to have a good solid reason why. So that's
here's an idea, and I think this is kind of general, but the Phenopackets would, in this case, there's not a primarily Mendelian disease, right? So the genetics fields of the Phenopackets would be a little bit less important. But if you have relatively rich phenotypic information, we could use those to run clustering programs. So for instance, we have a Python tool that takes Phenopackets and tries to predict how many clusters are there. Are there clusters at all, and if so, how many? And then we could sort of use that to say, does this patient have, I don't know, a differential distribution of cell types or something like that? We're actually working with Katie on or want to set up a collaboration. We should maybe talk and see if this might be synergistic.
That sounds very good. So maybe I should set up something with Richard and you. Have you talked with Richard Schurman, the scientific director?
A long time ago. I think he might even remember my name, but I haven't seen him.
We're connected. We're connected. Of course, he knows who you are. But we both worked for Craig Venter, so that's our connection. So he was the scientific director for JCVI, and I started the software group for Solaris.
We can talk about,
yeah,
or if
great.
anybody else is interested in this kind of thing. But yeah, I think that might be a potential use case, and we could maybe help with some of the software.
Sounds great. I'll reach out to him.
Excellent. This has been a lively discussion. So happy for all the questions and people contributing their thoughts and ideas and their use cases. But we're going to call it a day for the questions now, because we have a couple of last minute things for the last 5 minutes before we end. So first of all, thank you, ClinPheno people, for hosting me today, and for everyone who joined. I think we had 45 people online at the peak, so that's great. Hopefully you've learned something, you've connected with someone, you have new ideas for how to go away and develop and work on things with Phenopackets. But before you sign off, I kindly request that you quickly fill out this feedback survey. Beatrice, maybe if you could post the link in the chat. It's also linked in the slides here, essentially to ask how you thought today's AMA Ask Me Anything event went. We aim to keep hosting more of these for different standards or even different topics in the future, depending on what people would like to see. And in fact, Phenopackets was the most highly requested AMA after the Beacon AMA. So that's why we approached the ClinPheno group to host. So we do listen to your suggestions. And then as a reminder, so you can find a link to these slides and the agenda all through this document, which will also eventually be linked on the GA4GH website. We will also be uploading the recording of the call, and we will be uploading all of the questions that were asked, both verbally and in the chat, and we'll copy in the answers that were given over the next couple days or so, but also we'll share the link and ask if you think of an answer to a question a couple days from now, feel free to add it. It's sort of a living document, and this will also be linked from the GA4GH website. And we currently don't have our next GIF AMA session planned, but we'll take a look at your feedback in the survey. And if there's a standard that people really want to see us have one of these events, we will try to coordinate that. Or if you want to get in touch with me directly or Beatrice, feel free to also do that as well. So those, I think, were the last things I wanted to mention, and I'll hand it back to our ClinPheno leads if you wanted to say anything else before we close for today.
Yeah, thank you all for the great discussion. I would say please join our next Phenopackets meeting on June 12th, and we will be discussing what to prioritize from here, and in particular identify which specific implementation aspect we can kind of work on next together. And then our next ClinPheno wide meeting is June 18th, the following week. So I'd encourage you to join us there.